I'm Erica Hanson-Brown, and I'm your host today. I'm the founder and creator of One Cancer Place. My basic intention is to lead the way for more cancer patient recoveries. And there's a lot of work that we all have to do to see that, obviously. I want to give shout outs today to our founding industry partners, CGen Inc., and our supporting partners, which are the Colorectal Cancer Alliance, Buddha Logic, and Natera Inc. I'd like to introduce right now our guest host, Dr. Christopher Heery, Chief Medical Officer of ARCELLX Inc. of Gaithersburg, Maryland. Dr. Heery served as the Chief Medical Officer of Precision Biosciences and Bavarian Nordic before joining ARCELLX. Prior to his roles in biotech, he was head of the clinical trials group of the Laboratory of Tumor Immunology and Biology at the NCI, the National Cancer Institute. He joined the NCI Medical Oncology branch as a medical oncology fellow in 2009 and also held an adjunct appointment in the, Chris, you're going to have to pronounce this for me, genitourinary malignant branch. That's a stumble. At NCI, Dr. Heery was part of a larger effort of the Laboratory of Tumor Immunology and Biology to create new immunotherapies for the treatment of cancer. Dr. Heery is board certified in medical oncology and internal medicine, completed his internal medicine residency at the U of I at Chicago after receiving his MD from East Carolina University Brody School of Medicine. Dr. Heery, importantly, also holds a BA in English Lit from Duke University, and that explains his jokey nature. Dr. Flaherty, the guest expert for today, a gentleman that I am so in awe of for everything that he has done in his brilliant career. He is the Director of Clinical Research at the Mass General Hospital Cancer Center and is a professor of medicine at Harvard Medical School. He has made several seminal observations that have defined the treatment of melanoma when they established the efficacy of BRef, MEK, and combined BRef, MEK inhibition in patients with metastatic melanoma. So he knows a lot about melanoma, everybody. He is the principal investigator, the PI to us, of the NCI Matched trial, the first NCI-sponsored trial assigning patients to targeted therapy independent of tumor type. That's us, on the basis of DNA sequencing detection of oncogenes. Dr. Flaherty joined the NCI Board of Scientific Advisors in 2018, the AACR Board of Directors in 2019, and he serves as editor-in-chief of the Clinical Cancer Research. Take it away, Dr. Chris.
Oh, wow. Well, thank you for inviting me to be a part of the conversation, Erica. This is an honor for me to be able to talk with Dr. Flaherty today. I think as we think about what we want the larger scope of this initiative for One Cancer Place to be, we're trying to bring together patients who have an appetite for understanding some of the science with the current system that exists and helping bridge the gap for the patients that may not fully comprehend all of that science and need a bit of a guidepost that they can follow. So it is our hope that this series will be a starting point for people who want to be those leader type patients, who want to dig in a little bit deeper and understand some of the complexities of the system, some of the science, and how those things actually play together. So today, I think we're really lucky because we have a guest who has helped to navigate that and some of those treacherous waters very well in being in government situations like NCI related committees, but also founding companies that help to solve problems that maybe the current existing government entities don't solve for right away. So I think that's really exciting, and I'm really excited to get to some of his thoughts. Maybe to to start out, Dr. Flaherty, could you maybe just give us sort of broad strokes of, the last 20 years, everything you've learned? I'm just kidding. Where I'd like to start, honestly, from my observations as an external observer of things you've worked on is, seeing the ability to take a tumor that had essentially no therapeutic options and identify targets that were highly relevant, and then be able to create medicines that were relevant and actually helped patients considerably, and take that all the way from concept to an approved therapy that essentially anybody can get. Today, though, what do you feel? Do you feel that's been as successful as you would have wanted it to be? Like, are enough patients getting those therapies? Right. Or is the system still holding us back?
Oof. Wow. Well, first of all, thanks for the introductory comments. Very generous, both Erica, you, and Chris. So yeah, it's a big question. We'll probably unpack it in a few different ways, but let me just take a quick stab at it. 20 years ago and today, we constantly find a situation of new scientific evidence suggesting new starting points. I mean, let's just call them new potential targets. For kind of the beginning part of your question here. 20 years ago, really, the reason why I got into the field is that there was an accumulating set of evidence around the genetic architecture of cancer, like the mutations that happen in cancer cells that are the building blocks, that are the fundamental, what we call drivers of cancer behaving like cancer. Now, it turns out that actually every cancer cell beyond that also has some genetic alterations or mutations that don't have to do, that are just what we call passengers, like they're actually not responsible. And so, the decoding of the cancer genome, that was happening even back in the 80s to a degree, right? These tools that were developed scientifically actually were first put to use in cancer, dating back, to the early days of molecular biology in the 1980s. By the 1990s, that was accumulating even more. And so by that time, as I was considering career choices, it seemed like there was this accumulating body of evidence around the genetic architecture of cancer. And yet at that time, in the late 90s going into 2000, none of that had been used as starting points for new cancer therapies. And in fact, there was a lot of skepticism that you could actually, deconvolute the complexity of genetic information in cancer cells. And that, it's a kind of a signal to noise problem, that you might find mutations, sure, but but amongst many other mutations in a cancer cell, how would you decide, what to work on? So let me just use, kind of my melanoma experience and the BRef experience just to kind of quickly, kind of frame an answer here, which was that we had no notion really of a starting point in melanoma at the dawn of my career in the year 2000, but it was a clear unmet need area. It seemed to me that there were some scientific insights that weren't based on genetics so much, that suggested that what I refer to as kind of the wiring diagram inside of melanoma cells was beginning to be worked out. And in 2002, so kind of quite early in my career, BRaf mutations were discovered in about half of melanomas. And it turned out that, at least in melanoma, nearly always when you found a BRaf mutation, it was at one point in the gene and then in the protein that's encoded by that gene. So like a quote unquote hotspot, like a single point in that gene where these mutations happen. Well, we have 20,000 genes in the genome, and the number of actual, specific, what we call bases, like the DNA code, if you will, is, many millions that account for just those 20,000 genes, leave aside the dark matter beyond that. Anyway, what I'm getting at is if you just did the math, you'd say, this can't be an accident. It's not possible that this is noise as opposed to signal, right? But there are relatively few times in my career where like things are kind of that obvious. Okay, so that was published in Nature. It hit my desk or maybe screen at the time in 2002. Literally the day that news broke, I was like, okay, my compass is now pointed in this direction. But oddly, this happens over and over again, so I'm not patting myself on the back about this. It happens to everybody in the field. Like, that was my reaction. But nearly everybody else in the field, with the exception of two, are now close colleagues or became close colleagues at the time, nearly everybody else in the melanoma field says this is noise. This is not signal. right? I mean, the editors of Nature and the peer reviewers of that paper thought it was more than noise. And mind you, BRef mutations are found in about 8% of all cancer. So melanoma, yes, is one of the leading cancer types that has them, but they're distributed across cancer to varying degrees. Any case, so I literally kind of dropped everything and just kind of let my compass be pointed there. But again, even then, that wasn't clear to people at the time that this was, a clear signal. And so in 2021, we faced the same, kind of, I would say, set of opportunities and challenges, which is understanding, like, as we migrate from genetic sequencing towards other ways of molecularly characterizing, defining cancers, going way beyond what I refer to as site of origin. So in other words, breast, colon, lung, prostate cancer. We've worked to redefine cancer based on these genetic building blocks, but there's other layers of molecular information that we're just learning now how to use to define these starting points. And what I'm mostly getting at is that this is a constant problem, and patients are entering the stream. Well, it's moving. Where we're still trying to work on 20-year-old, what I would call 20-year-old ideas, and 5-year-old ideas, and then like fairly present ideas. And it's a lot. It's a lot for doctors. Scientists and doctors, to basically frame our, accumulated knowledge to say, okay, this is a fairly clear starting point. We somebody in our ecosystem, be it public sector or private sector, should devote effort towards trying to develop a real therapy here. But let me just kind of pause, in this answer for now and just, see where you want to go with it. But I would say first and foremost, the challenge that we've always faced in my 20-year career has been kind of prioritization. Like where are these, kind of how is it that we actually enumerate a high priority starting point? And if you rely on any one company, even a big pharma company that has lots of resources, or a small biotech company that's kind of got maybe like a single entry point, if you look at just one entity in the private sector, you're not going to get much of a global view, right, if you will, of like, okay, well, how do I boil the ocean of all these opportunities in terms of therapies that might move the needle, might cure me even, right? This is like, the stakes are obviously high when a new cancer diagnosis is confronted, but very rapidly one has to, as a patient, one has to actually try to understand, like, how does this world turn, and how do I, avoid chasing bright, shiny objects and actually gravitate to really high value proposition therapies.
Yeah. No, I think maybe to take one step a little bit back from that, I imagine for patients a question that comes up a lot is, well, I went ahead and sent my tumor off for sequencing, and maybe I had access to 2 or 3 services.
Yep.
And in this panel, I find this, and in this panel, some of them are the same, some of them are not.
Yep.
Is it worthwhile for me as a patient then to seek multiple different companies to sequence my tumor? And will that actually help me find a clinical trial that matches to me better? Do you have an opinion on that?
Oh, it's a great question, and this comes up. This is a daily issue. So what I would say is, well, let's again not skip over the fact that if you're diagnosed with a certain cancer type, and when I use the term cancer type, I mean that site of origin concept. So breast cancer, lung cancer, colon cancer. There are treatment options, right, that are well established that that have really substantial efficacy, depending on the cancer type, that shouldn't be ignored in this conversation, right? So understand that, like, not you, Chris, but like everybody on the call, today, or listens to this in the future, that's kind of the foundation we're starting with. Like, are there treatment options? Like, the answer is nearly always yes, or you get your quote unquote next generation sequencing or molecular testing / genetic testing, report handed to you. And this actually creates a problem right away, which I'm going to table for the moment and just now shift to your question. Right. So you're handed a report, and let's go with that scenario where you actually went ahead and got 2 or 3. What I would suggest, based on all my travels in molecular diagnostics, is that there will be consensus in terms of the genes that are being tested, and when I say consensus, so in terms of let's stick with that consensus on the genes that are being tested, when they point to therapies that we know are valuable, right? In other words, for lack of a better way of putting it, that like, improve overall survival, like allow patients to live longer, live better. So, clearly beneficial therapies. And so there's consent when we have those therapies, and there's genetic results that, point to those. I would say there's not variability to any appreciable degree on the testing of those. Now, the issue, as I alluded to before, is that cancer is complicated. There are multiple to many mutations per cancer cell. It varies depending on the cancer type, and then even individual patients with a cancer type. But in any case, where you will find variability is in the genes that are being tested across panels. Once you're down into the realm of, well, we're not totally sure, like, what the matching might be between this gene and a mutation in this gene and a therapy. There's a long list of those, hundreds, right, that we know are important in terms of cancer formation, like why cancer becomes cancer. That's why they're even on these gene panels. But their matching to therapy is what I'm getting at, is the uncertain part. And that's ultimately what people care about, right? I mean, the doctors, patients, everybody. And so that's where the confusion actually comes from, for the most part, is that a lot of those genes are of uncertain value in navigation. And you will find variability across those. And if that's what you see looking at 2 or 3 reports, if you're a patient and you're confronting this situation, I would say one's knee-jerk reaction should be, those are probably not decision makers. Like those, the fact that it's being tested at all on one panel and a mutation found in it, that's probably not a decision maker. Now, you need an expert to help you with this. I'm not suggesting that this is like, you can take my rule and say, okay, well, if it shows up on one report, not another, then ignore. No, because at any given time, we have, hundreds of therapies in clinical trials, and they're not all the same, which by that, I mean, there's some therapies that are in clinical trials, and dozens to hundreds of patients have been treated. This is a therapy that, in your and my opinion, is going to become an FDA-approved therapy, just a matter of time, right? So these are therapies that we use different terminology to, kind of flag them, if you will. But the FDA has this term breakthrough product for drugs that kind of show early results that look really promising. And if that holds up, this is going to be an FDA-approved product. But it might still be 2 years, even 3 years later before FDA approval happens. And so let me just use that as an example. If there's a genetic marker that is, yes, of uncertain match to an approved therapy, but points to one of those types of therapies, like, you want to know about that, right? I mean, like, absolutely positively, you want to know about that. I mean, if there are many other people in your same situation who have received that trial therapy and responded, right? It's like, you and your doctor want to deliberate on that. And so I'm not suggesting that, like, one should ignore everything that's in the investigational realm. Of course, I've made my entire career focusing on very, very early entry points in terms of therapeutic investigation, so I would never say that. But first and foremost, the thing to get right in testing is matching to therapies that we know are of value. And one should trust that that's happening, is what I'm getting at. Mm-hmm. Where it begins much more complicated is in this domain of the genes themselves, and then the specific alterations or mutations that are found, and whether those should point to what I would call a kind of high value proposition investigational therapy. Mm-hmm. And that's, I'm super unsatisfied with the state of the field regarding the, kind of the quality of the testing, the homogeneity of the testing, the standards, if you will, as it pertains to this domain. And that's where we need the most solutions in terms of decision support for patients and doctors, frankly. And in any case, I'll just leave that there.
Yeah. No, I think it is sort of that component, though, that we'd love to explore a little bit more, especially let's take an example that I think is close to, I know, Eric and I's heart, which is colorectal cancer. Right? So we have a disease that has had the same standard of care for almost 20 years now. And part of that is because that standard of care is pretty good, but it is definitely not great. And if you take it on a per patient basis, you have a pretty even, statistical distribution of outcomes across a population of patients who receive FOLFOX, for instance, including some who don't even respond to first-line therapy, of course. So I think where I'll speak for myself, but I think Erica agrees here, where we worry is that where standard of care is really well established, there's less incentive to get some of these molecular characterization of tumor done early on in the disease, which actually may eliminate windows of opportunity for some groups of patients where they might actually need those therapies, even investigational therapies. And so the question, I think, is what did going into founding companies help solve on that front?
Yep.
And what can we do as a group of patients and people who care about this stuff to try to push things in the right direction towards that solution?
Yeah. Well, yeah. How many solutions I've been a part of, I'm reluctant to suggest, but I've learned a lot to answer your question. And so, well, one of them is just kind of quickly that the wiring of our whole system between, initial pathology, molecular testing, so we call that molecular pathology, and then, kind of a tight mapping of that to therapy options, be they, FOLFOX, or, you're a colorectal cancer patient who has, like, I'll use an example from one of the companies that I founded, Loxo, which, the first drug we developed was an NTRK inhibitor, right? So, and I like to use that example because NTRK is these so-called NTRK fusions, where like a portion of the NTRK gene kind of moves literally and is stitched together with other portions of other genes. And in any case, those fusions are a theme in cancer. Between 5 and 10% of cancer are kind of driven by these fusions, and we've had recurrent successes with those. So ALK, ROS, RET, and NTRK, and now a few others emerging as we speak, have proven to be really valuable to know about. And that's why I want to stick with this, this kind of, answer of, as you said, like, why, as a colon cancer patient who faces the option of, FOLFOX as a first-line option, why is it important for you to have, testing right up front? And I would say, well, once we were some, months or years into development of that drug, it became a drug. It became literally an FDA designated breakthrough product based on what was running at about an 80% response rate, right, to a single agent pill-based therapy with like toxicity profile that was less than FOLFOX. Not that FOLFOX is so terrible, but anyway, it was definitely from a quality of life perspective. And these responses were durable, best we could tell at the, in these early days. They turned out to be quite durable with more time. And that's true of these fusion-driven cancers as a more general rule. They turn out to be relatively genetically simple. And so what I'm getting at here is that that's a nice, easy example to say, look, you want to know if whatever your cancer type is. So these NTRK fusions, I'm using them as an example because NTRK fusions are found literally across every single cancer type, but uncommonly, right? So like, in other words, on average, 0.2% of cancer, 0.2% of cancer, right? So it's a little bit of a needle in a haystack example, except that it's all over cancer. So there's no way you could say, well, this sarcoma patient or this breast cancer patient or this colon cancer patient is more or less likely to have an NTRK fusion. They all might have it. And then these other fusions that I was alluding to before, more variably distributed. But anyway, you get my point that you want to know. I mean, this is a test. It's it's rocket science, but it's not rocket science.
Can I ask you something about that? Because when I first saw the clinical data with the NTRK, my thought was, how did they run this trial? One, and second was, how is this ever going to be widely implemented around in the United States? Or I actually could imagine it in Europe more easily than in the United States. And I wanted to hear like lessons learned.
Well, we had to literally boil the ocean to find these patients. And, somewhere around the 30th patient or so that was enrolled on the phase 1/2 trial that became the trial that got the drug approved, we airlifted from Bangladesh. I mean, she happened to have testing with one of the standard commercial tests. That's a US-based company doing the testing, but it's, kind of globally available. So she was found to have one of these NTRK fusions, and her family and doctor figured out how to contact the team at Loxo, and we had to bring her to the U.S. It was for monthly visits basically to receive what was otherwise an oral therapy back in Bangladesh, and man, the amount of heavy lifting is, I'm just using that as an extreme example, but basically to find 0.2% of the cancer population and find handfuls of these patients to initially understand whether this drug might be worthwhile, and then to confirm that it was worthwhile. Yeah, it was hard work. So we partnered with basically all of the testing companies at the time that were, the big testing companies. We had some kind of relationship as a company with them, so that when they found one of those patients, we worked out a relationship where we basically said, look, you don't need to pick up the phone or do anything, just let us know, right? And then we'll do the work, right? We will reach out immediately to the doctor who ordered that test. And look, they might have already been put on FOLFOX, right? I mean, that decision might have already been made. In which case we would say, okay, don't stop the FOLFOX, but know that your patient has this, alteration for which we've got a drug, and let's say we were already dozens of patients into this, that's showing these results. Just FYI, this trial will be available, waiting, if you will, for this patient if they turn out not to be responsive to FOLFOX. And so this is kind of the purpose of this, this emphasis on getting frontline molecular testing is you may well go on to pursue therapy that has nothing to do with that testing, but you absolutely positively as a patient and a doctor want to know what is the thing in my back pocket, like that I've got my eyes absolutely focused on as my backup option, right? Like it's terrible. And I've unfortunately been in this situation many times when you're scrambling to try to figure that out once you know that FOLFOX isn't working, right? That's nobody wants to be in that situation. So so this is why testing upfront is so critical. Anyway, but trying to follow leads like that, like these kind of sparsely distributed, 0.2% types of opportunities. No one had done it before. There was a lot of skepticism about whether it could be done, like just kind of logistically and from a kind of cost-effective perspective in terms of taking investor capital and actually putting it to work. There really was a lot of skepticism around that point, but we had enough supporting investors to do the work. I would say, one of the things, we came out of that experience, I did anyway, hoping was that, okay, we've shown it, it can be done. And now they're actually, we know that there's a bunch of drug starting points or targets that are kind of like that, that are distributed broadly across cancer. Do we now see lots and lots of examples of companies, large and small, going after such things? Well, I wouldn't say lots and lots. We see some. There's still a lot of reluctance to pursue these things. And unfortunately, cancer is complicated. It's built on multiple to many different building blocks, and the permutations of those get even more complex in a way. And so we as a field, we need this to happen. Like we need starting points, large and smaller, frequent and infrequent is what I'm getting at. To be pursued when the biology makes it fairly clear that these could really be high value proposition targets. We can't have these kind of execution and ultimately market driven factors like paralyzing progress. And that's why at least we need to smooth out this hump that we're talking about now, which is is the U.S. cancer population, let's stick with the U.S. for now, undergoing molecular testing so we know who's who, right? Just so we even have that map laid out and can actually get information in the hands of people, even if they're receiving some other therapy, conventional chemotherapy, as a first line standard, fine. If it's efficacious, absolutely fine, for enough people. But you got to be able to look into the crystal ball a little bit in terms of what your treatment options are and not be scrambling, last minute. And it frustrates me to no end to be a part of conversations with, payers, insurance companies in particular, around this discussion of well, is it a standard to get next generation sequencing upon initial diagnosis of at least metastatic disease, if not earlier, or not? And so that it's still a debate drives me crazy. But I think the logic, and I think from an advocacy perspective, and you're all advocates, I'd say it's pretty clear. Even if you just only petition for one-time testing and just say at one point, right, at the time of initial surgical management of primary disease or upon first evidence of metastatic disease, at one time point, undergo this testing so you understand what your quote unquote genetic architecture and potential treatment options are.
So off of that point, I'm not going to try to solve every problem in the world, but I do want to point out one, which is that most patients in this country are going to be treated as their first stopping point at a community oncology center, right? And there is, to my knowledge, zero incentive for people that work at a place like that to learn about the complexity of the decision-making that you and I are talking about right now. They're not going to be reimbursed for the time it takes to read that report. They're not going to know the right network of people to reach out to get people into clinical trials. They're going to be faced with a lot of decisions in front of them all of a sudden that were a lot simpler, and it was just write the order for FOLFOX, right? And so I'm wondering, two questions out of this. One is, why did we need private companies to solve the problem of getting all of this validated testing done in a cohesive, unified way across this country, instead of why didn't something like Match help bridge that gap, and why didn't all NCI designated cancer centers have to screen every patient for everything? So that's one question. And the second question is, is there, and obviously you may not have an answer to this, but is there a theoretical policy approach that advocacy foundations could look toward
Absolutely.
to create incentives for community physicians to try to get involved in this type of work?
Oh, yeah, that's a really great way of phrasing that question. Make sure I don't mess it up in my response, because I'm going to start with part one, which is, kind of, effectively what, kind of some of the impediments to, having testing done in a widespread way. Let me start with the academic centers and then move to the community.
Okay, perfect.
And so basically, in the academic centers, you don't have to preach this to anybody. It's like that it's useful, sufficiently useful, let's say, right? Maybe not for all cancer patients. Are you going to get a result that's useful? I mean, and let me put some numbers to that. And the NCI match actually was quite useful for this purpose, because we sequenced all variety of cancer types, like literally every, from common to rare. We had a lot of rare cancers in the trial. We screened 6,000 patients, kind of centrally, which is not a universe, but it's a decent number. And out of that, we had what we saw, which was kind of predictable to a degree, was about a third of patients had a result that could point to either an FDA-approved therapy or an investigational therapy that we had in NCI match, right? So about a third of patients. And that was about a 50-50 split, actually. So break that third down into 1/6 and 1/6. So 1/6 of the patients, that testing basically either newly uncovered or rediscovered, because that patient might have already had some other type of testing before, possibly, that they have an FDA-approved option that's based on that finding, right? Okay, so 1/6, that's not a huge number. I'd like to be totally clear, right? But it's a number. And I think actually just about everybody in the oncology community, the treating community, would say 1/6 is enough to justify testing, right? I mean, 1 out of 6 cancer patients is going to have their treatment decided by this testing, pointing to FDA-approved treatments of known benefit. Yeah, we could all nod our heads.
I mean, to expand on that, we give therapies that we know only work in 1/6 of people all the time.
For sure, based on only knowing that they have colon cancer, exactly. And so in NCI match, we had a whole bunch, we had dozens of treatment options that we lined up that ended up being relevant for an additional 1/6 of patients. So, that we have a long way to go to get to 100% of patients for whom we can kind of find a molecular match, even if it's not, mutations, if we kind of agree to that. I'm just giving you a sense of kind of what we refer to as the utility of testing right now. And clear utility is pointing to FDA-approved therapies. I would make an argument that it's also useful to be pointing people to high-value proposition investigational therapies, as I suggested before. So, okay, so 1/6 plus 1/6, that gets you to 1/3. So, okay, so why is that non-valuable? In other words, in what setting is that not valuable? So let's start with the academic centers. Everybody in the academic centers would say it's valuable. We have we have large portfolios of clinical trials to try to basically match patients, in our own centers based on this testing, to at least 1/6 of the population, if not more, at any given time. And so, the issue is there. First and foremost, well, when we tried to do it ourselves, you asked the question, why do we need companies to do this? So at Mass General, I'm referring to here, we built our own test. This is not rocket science. I mean, it is and it isn't, right? But my point is, every major center in the country could do this because the technology is readily available and not owned by anybody. It wasn't, privately held intellectual property. In any case, we just lost money. We knew we were going to, but we were kind of hemorrhaging money as a system because we were doing the testing, offering it broadly to our entire cancer population, but payers weren't interested in paying. Now, mind you, we were doing this ahead of any other center doing next generation sequencing, and so we were kind of ahead of the curve. But we knew figured that was kind of our job, actually, to be ahead of the curve. And we lost money. We still to this day lose money doing in-house next generation sequencing. Now we're a nonprofit hospital, right? I mean, big academic medical centers, as a general rule, are nonprofit. Now, nonprofit doesn't mean that you can actually go into deficit, deeply year after year. So you kind of have to somehow balance the books. And we've been largely able to make that work. But other centers chose not to do that. In other words, not to voluntarily hemorrhage capital doing this type of testing. And so you found relatively few centers. I mean, really just a few major centers. I won't name names, just so as not to lead out others who chose not to do it. It's just a handful of major centers that decided, no, we're going to do it because it's just the right thing. So when that happens, you need some other solution. I mean, it could have come from taxpayer dollars. In other words, National Cancer Institute potentially. It didn't, in terms of how to make this kind of testing just broadly available outside of a clinical trial, single clinical trial context. And so when the answer is no and no to hospital settings, nonprofit hospital settings, and then NCI or taxpayer dollars, like what you're left with is the private sector, right? So that's this kind of process of elimination. In any case, so into that opportunity, I would argue to a degree, reluctantly stepped a couple of early entrants, into that space. Now it's well saturated, thankfully, right? So like these couple early risk takers, they were successful to a degree, and it then invited in companies behind that. This happens all the time, not just in diagnostics but therapeutics, and so the risk was taken, and to a degree, if you will, paid off enough. And now we're in an environment where 10 years later, this testing is considered something that we would call standard of care, and more payers. I mean, Medicare first and foremost, but then more and more private payers do pay for it, at least one-time testing. So we're a bit over the hump, but now let's transition to the community part of your question, because testing is not ubiquitous, right? I mean, this group of people and advocates know that already, that not everybody gets tested. And why is that? Now, patients can ask, obviously, right? They can ask what any of their providers, their medical oncologist or whomever, say, like, I want to get tested, and likely their response will be, okay, sure, we'll do it. But why is not every physician doing it, right? It's because of the numbers that I mentioned before, right? It's that more often than not, they're going to get a result back that doesn't point to a therapy. And in fact, I would argue it's worse than that. And maybe I'll kind of pause, my answer after this part just to see what I've left out, which is that depending on where you get the test, you will either get no support, right, in terms of what I call decision support. Like you'll get no support in terms of information regarding the relevance of that result to a matching to an FDA-approved therapy or investigational therapy, or neither. But there's very confusing decision support offered sometimes. The report will come back and basically it will perhaps say, nothing in the FDA-approved column, but here's a bunch of clinical trials you could consider, right? And if you're in the community and you're taking care of all variety of cancer patients, so you're not a melanoma expert, right? Like, as a melanoma experts, I would say, or any cancer-specific expert can generally digest this kind of information without much decision support and know, okay, these are the high-value proposition targets for which there are high-value proposition therapies, be they approved or investigational. But if you're treating all different cancer types, good luck to you keeping up with the kind of chaos of that information. And so this is where I think the real deficiency has been, that there hasn't been a model, public or private. And why can't this be public? I mean, I is my question, my persistent, more than decade-long question. Why can't we, put heads together? People like me have done this in nonprofit environments where we basically say, hey, look, as a melanoma expert, I will work with 9 other melanoma experts or 19 other ones, or you don't need so many, where we will all basically agree, look, these are the 10 genetic alterations that when we find them, right, we consider them to be must-have information because it helps navigate patients to therapy. You can gain consensus around that. Now, maybe after that, it becomes less consensus-driven because we just have so much less evidence, right? But in which case, I'd say it's not that important. And so, why is it that we still are struggling with this problem? Until we solve this problem, if I were in the community, I have to say, to be handed a report that is at a minimum confusing and a maximum, just a chaos generator, right? In terms of the amount of follow-on work that's now created that isn't more, many more times than not doesn't lead to a satisfying result of a patient actually being matched to a therapy, in a trial or not in a trial. You'd say, I just as soon not get the information. I mean, that's the decision that people are making. Of course, you know that I'm not saying that I would ever, those words would ever come out of my mouth, but I've had so many of these conversations. I understand the problem. We just need to hone a solution somewhere between the public and the private sector. Just to finish up on this point, well, if it's such an unmet need, why wouldn't it just be jumped on as a business opportunity? And that's because decision support, right? This thing that I'm referring to, that's not intellectual property. Well, it can be, but it's not. As a starting point, it's not a highly valuable commodity that one can package up and create a product around and sell. I'm not saying it's of no value. That's not my point. But it's different than a therapy, which is highly valuable and well reimbursed, and then the testing, which is not reimbursed as well, but is still sufficiently valuable. Now we're down to a level where the market doesn't work in terms of creating incentives. That's my final point there.
No, I think it highlights exactly where the gap is, that one cancer place is trying to
exactly.
And I think this problem, it comes back to this question of how could we create something to fill that gap? What does that look like? And I don't know that we're going to come to an answer today. That'd be great. But I think as long as we, you can't solve a problem until you've identified the problem. And I think it's important for this group and anybody listening in the future to understand that this is a major problem. Most medical oncologists in the United States have no idea how to manage the results that come back on one of those tests, unless it is an absolutely clear, if you have this mutation, you should take this drug, right?
Yep.
Outside of that, and even then, they might have a hard time getting access to the drug because they haven't used it before, blah, blah, blah. Yep. But if it's picked between these 30 clinical trials with completely varying levels of evidence behind them, most people don't know what to do with that information. And that includes, I don't want to speak for you, but includes someone like me who spends 100% of their effort picking clinical trials for patients. It's hard. You look at these lists and they don't make some of them don't even make any sense. And you would have to know something about that drug to know that it doesn't make sense. So it's a tough problem, but definitely a gap that this group could try to fill and brainstorm and think about, like, how do we solve that problem? Could there be a service, for instance?
Oh no, for sure. To me, this is exactly what your group is aiming for. Is to fill this gap. Look, some people do it by getting on the airplane and flying to 10 different, centers. This drives me crazy, right? That the amount of resource and anguish and time that, people expend, to do that. It doesn't take that is what I'm getting at, right? This is a solvable problem. So if you put, if you're a newly diagnosed metastatic colon cancer patient and you enter that into clinicaltrials.gov, which is, let's like celebrate the fact that at least we do have this NCI supported, taxpayer supported, kind of repository of essentially all clinical trials. It's become a requirement that even phase 1, early phase clinical trials, but anything beyond that as well are, registered in that database. But if you put metastatic colon cancer in and nothing else, you'll get more than 500 results, right? In melanoma, the number's about 500, but for colon cancer, it's probably 1,200. Yeah. Right? It's unmanageable, right? Completely. But it turns out that right off the bat, 700 of those out of the 1,200, they have actually nothing to do with colon cancer, as it turns out. It's just that they have to do with any cancer, and the trial's open to any cancer patient. And if colon cancer is any cancer, so therefore it's included. So there are ways to parse these lists, but let me not build from the bottom, which is that, and kind of more shift to the top. Okay, FDA-approved therapies, we can all agree that like those should be discussed. They're approved for a reason. Then come these drugs that are actually within months of FDA approval, right? They're being made available through expanded access, what we used to call compassionate use, but that term's gotten more complicated lately. Expanded access is this, bridging time of, it's on average 6 months, but can be even up to a year, where the evidence has already been generated. The drug's going to be FDA approved. Like it's essentially certain, and companies will make their drugs available bridging to that moment that the drug's FDA approved. You want to know about those therapies if you've got a cancer type and or molecular type that points to one of those. So that's like adjacent to FDA approved therapies. Then come those breakthrough products that I was referring to, where there's already evidence, and the FDA, like the same body that reviews and approved drugs in their full, data set, has already said this looks really, really promising. And the conversion rate of breakthrough products to FDA approval is inordinately high. So my contention is you also must know about those, right? Yes, they're still investigational, right? So in my worldview, you must know about those. After that, it becomes shade of gray in a way, right? I'm not saying that there aren't therapies that doctors and patients should know about, but if we could just solve the first part, right, we'd be a far better place than we are right now. And actually, everybody would then understand, this is why we're doing the testing. This is why we're, honing our decision support strategy as an entire community. It's because we don't want people to miss out on anything that I've just mentioned so far. It's much harder work to a degree, but it's work, as I said, that you find plenty of oncologists, certainly those in the academic, centers who do this work, who, volunteer their thoughts all the time. Where patients don't have to come see us. We can bottle up and, kind of mash together our thoughts for what we think are the most compelling, right? So I'm using that term as opposed to any of the other terms I've used so far. investigational therapies that are based on some really solid science. They look really quite promising, but the human evidence is either just emerging, right? It's too soon to really make a strong call, or maybe it's, even earlier than that, just too few patients, very early, but it's in clinical trials. And considering that patients, treatment options otherwise, there are times, certainly if they've tried FOLFOX and tried FOLFIRI, combined with another agent and perhaps even a third line option for colon cancer. So now you're talking about fourth line therapy for colon cancer.
Right.
At that point, a lot of things in this, what I'm calling shade of gray category, are certainly should be discussed as the next most valuable potential therapies. Now, look, this becomes a situation of where you're being cared for. Are you going to have to kind of commute to a center that has these clinical trials? We're doing a terrible job, although we talk about it a lot in the field, of like bringing clinical trials to patients as opposed to having them have to come to major centers. That's its own conversation. But in any case, my point is that it is part of the discussion, right? This is not like a just walk down to your local pharmacy, and kind of get access to investigational therapy. But nonetheless, this is the hardest work. What I'm talking about, it's going to take the most kind of engineering of our community to ultimately solve it and make this kind of expert opinion broadly available to everybody. But it's doable. We certainly have the technology to do it. But I just want to be clear that it's the kind of the muddy waters that I'm referring to and clarifying those. We're not talking about therapies that we know are valuable, right? I mean, we're talking about therapies that may turn into the next breakthroughs. I mean, they may, based on what we are looking at in terms of the preclinical sort of translational evidence, but they're not there yet. And so, yes, I see many patients who want to know about exactly those options and the best of those options, no question. But I think we have to solve from the kind of top down, if you will, in terms of how we build these communities.
That makes, unfortunately, that's the only model that's worked so far. And I think, it's incremental, but I wanted to ask you one last thing and then see if anybody has any burning questions, which is, I think there's accumulating evidence in addition to sequencing the genome of cancer cells that looking at the expression of certain genes within those tumor cells is actually highly relevant in a pretty large proportion of overall cancers. However, it appears to me that RNA-seq hasn't, or any gene expression profiling you want to use, hasn't quite made it to the same level in a lot of these panels.
Yep.
Even though it may be more important in some of the bigger cancer types, because what we're actually seeing is that like colon cancer is probably 20 or 30 different diseases. It's just, we've always called it all the same thing.
Yeah.
And for every person, you could make the argument, each one is actually a different cancer. But as we move more towards our understanding of not just the genes themselves, but how they're expressed, and that may be dependent on the tumor tissue or the tissue from which it derived. Do you see that coming to the forefront?
Oh, absolutely. But what a crazy backlog. I talked about the backlog of just kind of, leveraging the genetic, the mutation information. It's an even bigger backlog in the domain you're referring to. In other words, at the dawn of my career, when you went to AACR, the biggest, science conference in cancer, just like talk after talk, poster after poster was RNA expression analysis of tumors. Like that, you just see 10 tumors, 10 colon cancer, two individual tumors from individual patients, being sequenced at the RNA level, and that data projected in chaotic, confusing ways. And so the technology, my point is, has been with us for even longer than that, but certainly even applied to human tumor samples, it's a 20-year-old, technology, and we barely tapped into it. I mean, just barely touched it. I mean, you've worked in immunotherapy a good bit of your career, and I would argue it's actually, like, if you were to say, like, where in therapeutic development could this be most valuable? I'd say we already have enough evidence to say that not just for the available immunotherapies, PD-1, CTLA-4 antibodies, but next-gen immunotherapies as well. I'd say, well, it's an RNA sequencing, not DNA sequencing. It's RNA sequencing, and not hunting for mutations, but looking for these expression patterns. This is absolutely staring at us. We have other technologies that are emerging rapidly, as in proteomics, like right behind RNA sequencing, that are similarly going to, offer us opportunity for better precision medicine concepts, for available therapy mapping as well as investigational therapies. So we are so behind the curve on the RNA sequencing side. It's really kind of crazy. there are some testing platforms that actually, when looking for as part of their mutation seeking process, do RNA sequencing. And so it's like quietly being done in the background, but it's not being used. And this is a twofold process. First of all, there's a lot of data that's already been published, scientific data published by research institutions, including mine, that basically show really striking relationships between this RNA expression and outcome with even chemotherapy, but certainly immunotherapy and certain targeted therapies. And so validating that and actually having that become part of our precision medicine platform, like we're behind the curve in doing that. And again, the market's not solving this problem for us. It's another real inefficiency. But there's a whole generation of therapies coming that my view, they're going to be best navigated by this concept, this platform. I'll just finish by saying there are complexities to this. I mean, this is, in other words, more often than not, when you talk about RNA sequencing, you need to pay attention to a handful of genes and their expression, not just one, and not just a single alteration or mutation, as I was referring to. So when it comes to mutation calling, it's not a binary result world, but it feels like a binary result world compared to RNA sequencing. And that does create challenges in terms of turning those into diagnostics, locking them down, having them ultimately become FDA approved and paired with the therapies. I'm being fast about this and answering it, but basically just saying that there is a hump to overcome there. And I and many others in discussing this with FDA, who again are they are public servants, they're part of the same community that we are. There's a concerted effort to try to create a roadmap that makes it very clear and not so intimidating for companies, frankly, to enter into this domain, because the backlog, I'll just emphasize, is large and growing larger.
Yeah, well, I'm glad to hear you say that. I mean, it is I think people tend to think of these technologies as competing. I don't think they're competing at all. I think they're complementary. And
totally.
I think the challenge is that there are some really great potential therapies out there that have been created to target pathways that we know are overexpressed in certain cancers, but the companies don't know which patient is which because nobody's testing. So it's a bit of a vicious circle here.
That's right.
I'd like to make sure that everyone is aware that you can post a question in the chat, or if you'd like to come off mute and ask a question, you can go ahead. We only have about 5 more minutes, and we want to make sure that Dr. Flaherty is able to go on to his next meeting. I'm sure he's busy after this, but so if anybody has one, go ahead.
I have a quick one. I just have a quick one. Dr. Flaherty, if I were a defined BRef patient with any organ site, would I not be smart to come and seek you out as a treating physician?
Well, yeah, I objected before to the seeking out part. I mean, getting on an airplane and coming to see me is not the way that all cancer patients should have to navigate their treatment options. I mean, so I'm using that directly referring to me, but I mean that for any pairing of cancer type with molecular alteration. There's a world's expert out there, or a handful of them. And what the information should come to you, you shouldn't have to come to the doctor. I'm just as a starting point. I'm not saying that that every cancer patient shouldn't have a conversation. In addition to just getting the information. But in any case, start with that as a first response. Right. No, so I think it's, there's a community of us. I focused on melanoma as an unmet need area in the beginning of my career, and still to this day, that's my cancer type, focus. But I also I primarily got into the cancer field to try to address this overall problem of therapeutic unmet need. And my interests are broad across cancer. That's why I built a research group that covers all of cancer, but it's made up of people who focus on different cancer types and different molecular, subtypes, if you will, subpopulations. So even at Mass General, there's a handful of us who focus on BRef mutations in their various contexts, right? As you know, about 8% of colon cancer patients have BRef mutations. That's its own group and own group of patients. And, my close colleague at MGH, Ryan Corcoran's a world's expert in that topic. And we put our heads together all the time in terms of clinical care and research questions. And in our lung cancer group, we have Abai Dagogo Jack, who's a lung cancer expert who focuses on BRef mutations in lung cancer, where about 4% of lung cancer patients have them. And so what I'm getting at is that as more time has passed and these areas of research have become more mature, you'll find experts, kind of popping up here. And to your question, I think navigating to those experts, at least electronically, in other words, by email communication, kind of getting, quick, input of like, I've sourced these treatment options. Does this sound right to you? These aren't challenging really questions to answer. Again, ultimately there needs to be an oncologist who obviously is, someone who any given patient can have a full, discussion of all the treatment options and, all the considerations about logistics of therapy and impact on quality of life and what can that patient themselves and their family manage and, so on and so forth. But getting access to the information you're talking about, I think is the big gap. We just have to find more efficient ways so that everybody can get that.
So Brad had a question too, which I think is a great one maybe to end on, which is, you use sort of an entrepreneurial spirit to try to solve problems you were seeing in the oncology space. Was that a means to an end, or did you just think it was the most efficient way, or what? How did you go down that path?
Well, both. I mean, it's most efficient in the sense that in the public sector, there's just some things that either won't move or will move at geologic pace. I mean, right? There's just too much of a hump to get over. And so if there's a way in which you can make a pitch for a solution in the private sector, sometimes that's simply the most efficient way to get to, what we'll call proof of concept, like that, proof that this idea, might be effective. The means to an end, I mean, this has been my version of why I've ever put my foot partially in the private sector, is to then create a solution that then will be stolen and multiplied and ripped off and used by whatever number of companies that want to be fast followers and promulgate this approach, this concept, right? Whether it's a therapeutic concept or a diagnostic concept or decision support, I don't care, right? So this is like the idea of owning any of this has like never been my ambition. And so obviously investors want returns, I get that, but there's a way to do that math. Right, where you basically you demonstrate, you make it look easy is what I'm getting at, right? Like the thing that seemed impossible or hard, you make it look easy, and then everybody nods their head and said, "Oh yeah, like we can do this," right? So that's the means to an end. And for everything we've talked about today, these things will happen. And what I would just encourage you as a community to consider is, as advocates, work with the public sector is of course NIH, NCI, academic centers, right? That's all public sector, but there's obviously many constituents there. Work with the private sector also. I mean, and then sometimes tying all those together, it becomes then, kind of the triumvirate that's needed to ultimately land on, starting points for solutions. Once the solution has been demonstrated to work, that someone will actually reduce that to practice and make it, a business. Trust that that'll happen. It's this getting over the hump piece. That's the hard part. And that's why I encourage anybody ever in my world, certainly, to, kind of toggle or step into the private sector for, in a kind of part-time-ish way to just push towards solution creation in that domain, because time and time again, I've seen it. I mean, I just watched it passively where, once it's been demonstrated once that it's workable, then kind of all downhill from there.
I think that's a great note to end on. Erica, do you want to have any last comment before we finish?
Let me just thank Dr. Flaherty for his enthusiasm and passion for the patient experience. We're so honored that you joined us today, Dr. Flaherty, with your background. I think you just described the answer to one of my questions, which was, the layman's answer to what is translational medicine. You embody it, and your answer just explained it. And I'm so proud to launch our series, our autumn series, with you and your passion really for the patient experience and the patient recovery from cancer. So my personal thanks to you, and I know we'll be talking soon, and I'm looking forward to it. It's an honor to know you.
Thanks so much, Erica.
Very much.
And thank you, Chris.
Yeah, this was so fun. Thanks for having me along. So I think we will let Dr. Flaherty go.
Let him go, poor guy.
All right, take care all.
All right, thanks.
Thank you so much.
Bye.
So everybody, what we're planning on doing is, we hope that there might be some questions, some follow-up questions that you might have had and might continue to have, to just ask Chris, Dr. Heery. Since he's a lit graduate, he's really good at explaining things in simple ways, right, Chris? And let's have at some questions, because I'm sure everybody here is so smart about your experience and the cancer treatment landscape and everything. And I know that Chris might be able to explain some of what Dr. Flaherty talked about or other things. So have at that if you'd like.
I have a question that more came from what he said, not what he said. But I've kind of envisioned it as, hey, we've been very cancer type focused, but as we learn more about mutations, kind of as I envision treatment in the future, it would be more maybe mutation driven treatment. So like if you're BRef, V600E, then it doesn't matter if you have colon cancer, you have breast cancer, you have whatever. But then you look at some of the trials that were pan-cancer, and the impact of the treatment varies by cancer type. So you kind of go back to cancer type. And then when he was talking, he was saying like you take, the top 9 people on melanoma and you kind of come up with what is that path of the options. Yeah. So I'm just wondering if that's like a pie in the sky idea that, hey, that's where we'll eventually be, or if that's actually not how it works, that it really has to be the mutation in context and things will, you know what I mean? So it just surprised me, because I didn't feel like that was kind of down that path.
Yeah, we kind of ran out of time. That was going to be my next question when we talked about RNA, because so if this isn't something everybody knows, I'll just say this quickly. You've probably heard a lot about RNA in the last year and a half because of vaccines, but DNA is basically the code that is going to carry with us in every cell of our body, right? And from the DNA, you are going to then turn some of that by transcription into RNA, and then RNA is going to be translated into proteins, and proteins are what make our cells do whatever their function is. But in a cell, in your skin versus a cell in your liver, different genes are going to be expressed differently, so that they make different sets of proteins, so that those cells do different things, right? So a mutation in a gene in a liver cell might have a very different effect than the same exact mutation in a gene in a skin cell, right? And so, for instance, a BRef mutation in a skin cell, particularly a melanocyte, almost 100% of the time is what will drive a melanoma. That's what he was referring to. But that same mutation in a colon cancer cell might just be there by accident, or it might be playing 20% of the role of why that cell behaves the way it behaves, or some number, just pick a number, right? So what that means is, if you treat every patient with melanoma with that BRef V600E mutation inhibitor, you're going to get a response in the vast majority of those patients. And in colon cancer, that number is going to be much lower. And it is that context that when targeted therapies first were being developed was a little bit lost. Like, oh, it's not going to matter what cell this thing came from. And in certain very specific situations, that is true. But in most situations, it is not true. Not totally true. I don't want to say it's not true. It's just not 100% true. And so what that means is you do need the context. So I don't know that we're ever going to get away from saying, what is the cell from which this cancer derived? Because that's relevant to the mutation you find inside of it, right? And that's where that complement of DNA sequencing and RNA-seq is really going to play a part. Because if you find a mutation in the gene, in the DNA, and it is not expressed in RNA at all, well, then inhibiting it makes no difference whatsoever, right? But if you find that same mutation and you find that now it's overexpressed aberrantly or incorrectly, then inhibiting it's going to make a ton of sense. right? So I think that's just a good example of how these technologies are going to play together. And I think what Dr. Flaherty was describing was, this is how we got to this point in this field, by identifying gene mutations that are drivers. And what he was saying is they found that in melanoma. So of course they knew that there was far less noise when every time it was the exact same mutation in that disease, right? But I don't think that we're ready to say that about all cancers for a given mutation. I think that's a little bit of a bridge too far. I'd love to get his opinion on that. I wish we had a little more time with him, but maybe I'll follow up with him over email and ask that question, on his opinion, because I think it's really important to get it from, somebody who spent 20 years working on this particular problem.
Where I was coming from on that, my question to him was my experience in talking to patients and recommending that they find the doc that's passionate about their mutation, whatever they found is their mutation, that's a wise thing for a patient to do. And I was going over that bridge too far in saying, well, since I like you and you specialize in research on BRef and I have BRef in colon, wouldn't I be smart to come and see you? But what you're saying is that really, and what he recommended, if I heard this correctly, was no, it's probably better to go to Scott Kopetz, who specializes in colorectal and loves and is impassioned about studying and finding answers to the BRef mutation, probably pathways therefrom and all that. Am I correct on that, Chris?
I think, and I hate to interpret other people's words, but I think what he was saying is it's not fair that people with resources can go and try to see him if they have a BRef mutation, and 90% of people out there, or 95, 99% of people cannot. We face this as people in a field that know a lot about one particular thing a lot, right? Like people want to come see you about this one thing. But what probably a better use of everyone's time is to find a physician who cares and is willing to learn and listen, local to you, and then set up a bridge of communication with an expert. Right? Mm-hmm. So for instance, I have a friend who had testicular cancer years ago. The world expert, the person who invented the therapy, the combination chemotherapies that cures testicular cancer at extremely high rate. He's a guy at Indiana, super famous in our little world. But you can email him and he will email you back 10 minutes later. I mean, if I emailed him as somebody he's never met, he emailed me back. He's like, hey, great question. Here's what I think, right? And sometimes it'll be 100% of what you need. Sometimes it'll be 50%. Sometimes it'll be 75%. But it is very rare for somebody in an academic world to get an email from a treating physician anywhere and not want to answer it. It's the whole reason you work in academic medicine. One, because you care, and two, because you stuff that other people don't know, so you can tell them stuff, right? So it's just, I think what he was getting at is the treatment and the good decision making should come to patients, and we need to help facilitate that a bit more. And that doesn't mean you need to get on a plane and fly to them, but it does mean you need to find a way to communicate with them. Now, if that means you have to drive 20 miles to go to your closest academic medical center, or 50 miles, that's worth it, even if you're not going to keep going back. But every time you reach a major decision point, you have that communication with somebody who can talk to the world expert for you. And I think some patients, they want to be in that loop of communication, but it's really inefficient. It is super inefficient to do it that way. Because, we're trained to use certain words and things that, frankly, we don't want to use in front of patients because sometimes they sound kind of harsh or cold or whatever, but they do help us communicate quickly. Mm-hmm. So there are real advantages to finding a doctor who can do that for you, local to you. It doesn't mean you can't go meet them once, maybe, or something like that. Like one of my favorite physicians is John Marshall. He's a colorectal, but other GI cancer specialist. But the reason he's been so successful in doing what he does is that he only sees patients once if he can, because he would rather not steal patients from the community oncologists, because he knows they'll stop referring them and he won't be able to give good advice to the patients anymore. Right. So by only seeing them once and then sending them back and then being very communicative with their physician, he actually improves the quality of care for more people overall, right? So I think that's what he meant, although, I don't want to put words in his mouth.
It sounds to me like it's coming from out of compassion. And
Yeah,
I think we all appreciate that a great deal.
I think we just had the great privilege of talking to somebody who over the course of his career will improve thousands, hundreds of thousands of lives, not just through developing new drugs, but trying to create systems that actually improve the way we make decisions. And that is not always appreciated, but it is a very important component to oncology care. It's critical, right? I mean, you cannot get the right treatment without the right inputs and the right information. And he's trying to create better information systems. I have to thank you again for letting me have that conversation, because as a nerd of this stuff, it's really fun to talk to somebody who's already lived it.
Oh, you help us so much, Chris. I mean, we know, and I think I speak for all of us, enough to be kind of dangerous. I know that I'm kind of dangerous, but we all want to know more. And I'm speaking for all of us again, but I know that I'm passionate about knowing more and knowing why and knowing how, and wanting to know the best way to help other people find their way through this very, very difficult system. So I'm really glad that you stepped forward and said you'd do this, because I didn't know what I was going to ask him, because he's so brilliant. And I feel like I know nothing, even though I know as a patient, I know a lot. But he made me feel like I know a lot. And I think everybody else agrees with me on that. And, I just think we're so honored to launch this whole thing with someone of the high quality and caliber of somebody like both of you, Chris, honestly. And I'm sorry, I should call you Dr. Heery and treat you
No, you shouldn't.
that you deserve.
No, it's fine.
But does anybody else have any more thoughts? I bet you do.
I have another question, if nobody else, because I don't want to steal all the questions, but
Go for it.
So something I just think he raised an interesting point in terms of kind of the burden of information sometimes for physicians who get these reports and they're like, what do you do? You know what I mean? Like, because it's not clear. And so he was talking about that from a helping in the decision-making of physicians. I think what we struggle with is helping with the burden of information and kind of navigating that for patients, right? Because I think there's kind of a couple different things at play. One is sometimes physicians don't tell patients things because it's kind of irrelevant and they don't really need to know. But then it makes the patient wonder if they're being left out or their doctor doesn't know or whatever, kind of thing. And sometimes it's like in the best interest, but they don't necessarily go through that and say, well, you have these 5 mutations, but they're totally not actionable. So I'm not even going to discuss them with you because, it's kind of, burden of information. So how do we think about with patients, the idea of, NGS testing is really, really valuable, but you have to know what's valuable about it and when it's valuable about it. And I think that's what he was saying in terms of physicians, but it's also true for us when we're talking to patients, get your NGS testing, and then people get it back and then they freak out and go, what do I do? And so what are your thoughts on that in terms of how to encourage patients to become informed, but also help them navigate when that information is helpful and maybe not?
I honestly think this is a huge challenge. I will tell you, I do not know the answer to this or even really have a great idea, other than to say, I think part of what makes those reports so confusing is that it is not super obvious what is the thing that is obvious and you should absolutely do this, and what is the thing that is just like, we're just brainstorming here. Like, I really think they should make a picture or something that's with a little thought cloud for the ones that are just like, hey, you could try these clinical trials, right? Because they are just guesses. They are just guesses. And I mean that as negatively as I can mean it. I don't mean that like, oh yeah, they're doing their best. I mean, it's just these are random word association from an algorithm. Okay. They're not good. Some of them are better level of evidence. Like at least there's a phase 1 with some patients who've responded. Well, okay, let's make some sort of color code, like something that makes it obvious that this is different. And like a link to where can you find this data? Like where can I find something like just a press release or something that tells me this is real? I don't see why that would be hard. Now, I'll grant you, I don't look at a lot of these reports anymore because I'm not taking care of patients anymore. But I'll tell you, 5 years ago, those reports, when I would get them, I would have to spend 20 minutes looking at it to make sure that I wasn't confusing what was the intent of the report. So I think the number one thing is that a group like this could be advisors to Tempest and Foundation and Caris and whomever and Strata, and say, look, here is what we need to see so that this makes sense. And like they should actually sit down with people and say, if you were handed this, would you know what to do with it, right? And frankly, they should be doing that with physicians. Like that should be part of the business model, right? They should have an advisory board of physicians, not physicians like me or Dr. Flaherty. Like and granted, he would know much more what to do with that than I would, but it should be like Joe Schmo oncologist in Kansas, right? I mean, that's who it should be because they're the one who has to deal with it. So I think there's a flaw. Like, I think it's still too early that nobody has received that sort of critical feedback and started saying, I'm not using that one because they have a bad report. I'm using this one because the report is easy to understand. The market will usually drive that, but most physicians haven't leaned into reading any of them in the first place. So they don't, they don't know, but over time they will. I'm sure they will.
I was going to say, this might be a really dumb question, but why do the reports, why are they so different, right? What process are they using that makes the report so inconsistent from one report to another?
Well, I think Dr. Flaherty touched on this just a little bit. I asked him this question in a slightly different way, but I think what he was saying is each one of those companies doesn't know which thing is going to be the most relevant, so they're all making their best guess. So for anything that's not obvious, everything after that, either they have the right primer, they have the right assay already set up, whatever it might be, they've already made a bet on it. And it might be because they're looking at the therapeutic landscape and saying, I want to have this in here because I think this one's going to work later. And so I want to be able to test this on hundreds of thousands of people now so I know what the incidence is and I can sell it to this other company who's developing the drug. Or in the case of like Foundation, right? Like they're owned by a drug development company, right? Mm-hmm. So like,
they have
you can bet that they're going to enrich in their panel for the things that are relevant to the drugs they're developing, right? Like, and not to make it sound evil, it's not. I mean, this is part of the necessity of drug development, but there are incentives for each company to have different things in their panel.
Chris, do you think there'll ever be the day when somebody will insist that all this information becomes standardized?
Oh yeah.
When?
I mean, I think we're close to that day. I think we're close to that day for the things that we already know, right?
Yeah.
The problem is, I insist that RAS mutations in colon cancer are standard. How much effect does that have in everyday real world medicine? It not enough. I can tell you that much. It's like 50% of community oncologists check for RAS mutations in colon cancer. What?
50?
That's something we absolutely know matters, and it's not done. So imagine what the inertia is, or like what the starting energy is to get these really new things into the hands of treating physicians all over the country. And listen, I am not saying this to disparage community oncologists. I'm saying they're trying to take care of a person with breast cancer and then a colon cancer and lung cancer, like in order. It's really hard to do that even with the current standard of care, let alone next generation stuff. So somebody has to create systems that make it easier for people in those positions to know what to do with the information. Or we have to, like this group would say, you have to see people who know what they're doing for my cancer in particular. And I'm not going to go see someone that sees breast cancer and then colon cancer and then lung cancer, because I know I'm not getting the best quality outcome. But I'd say we're a long way from that, Erica. I mean, wouldn't you say you talk to a lot of patients? There are a lot of patients who are like, well, I like this doctor and that's good enough for me. Right? I mean,
yeah,
so,
here, this raises another question that I had, because he was big on talking about the gap. You guys both were in decision support. And my question really is the second opinion. I know that's different than decision support, but might a stopgap measure be an insistence to, or a recommendation to these brand new patients that at the very least they seek out and we help them get a second opinion right at the from the get-go, and then start developing that conversation about and utilizing decision support and some of the stuff that they develop? Thanks. I wondered about that.
Totally. I mean, I think what his point was is if every patient with a BRef mutation tried to come see him, there's no way, right?
Well, right.
Right. But if you go to see, if you live in Wisconsin, the University of Wisconsin is a fantastic medical center. Yeah. Go just once, right? And then if the recommendation from the GI expert at University of Wisconsin is the same as your local oncologist in wherever, Green Bay, then just follow that plan with that oncologist. It's super simple, right? And we say the same thing. In North Carolina, we have 3 top-tier medical centers. You don't need to go to them all the time. You just go there to find out what's the hard decision to be made, right?
Yeah.
And I think that's a much more sustainable model, but I do think if you're asking what should one cancer place and what should this community foster, it should be, if you have a newly diagnosed triple negative breast cancer, you need to go see a triple negative breast cancer expert tomorrow, right? Find out what their recommendations are. And if they match with the oncologist you want to see because you like that person, you can go to that person, no problem. But if they don't, you might want to think very seriously about why they don't and what you want to do with that information. Then I would say third opinion, right? That's the only time I recommend like a third opinion is like if you have if you get the same exact information from 2 doctors, the chances of that are very high because doctors almost never agree on even the most obvious things.
Yeah.
So that would be absolutely my recommendation. Yeah.
Or I think too, the idea of treating physician versus consulting physician. So, you and Dr. Flaherty both are talking to that. And I think that for patients, that mindset of just because you're getting a second opinion doesn't mean you're changing doctors. You can still be treated by your physician, whoever it may be. And if you have a great relationship with them and they're great, I think the question for them is, will you work with a consulting physician who's a specialist in this area? And if they say, yeah, and they're willing to, and they can, so, and the treatment is something that they can offer, or they're willing to have you go someplace else to get that treatment, I think that's the ideal scenario. Because then you have a physician who's close to you, who you really have a good rapport with, but who has that consulting relationship with somebody. And that I think is ideal because then you have somebody like Dr. Flaherty who can't see every BRef patient, but you have your local oncologist that will work with Dr. Flaherty, and that I think that to me is more is your oncologist willing to work with consulting physician or consultants, and then get a second opinion that's a consult. Because I think a lot of people feel like getting a second opinion means they're deciding whether to stay or not stay with their doctor. And that's really not what it's about at all. It's about getting expertise. And so I think reframing that with patients and kind of mapping that out for people so that they go, oh, you can do that? Like, oh, wow. That I think is a more beneficial to the system and like the effectiveness of the system. And then also for that patient to be able to, to feel comfortable in that choice.
I think that is fantastic advice, right? To me, there's only one red flag for doctors. It's if they say they know everything. No, you don't. I can guarantee you don't. And if somebody believes that they can make every decision by themselves without asking any other advice, huge red flag. And if somebody says, don't, if you go see a second opinion, I don't want you as my patient. Well, good news for you. You don't want them as your doctor, right? Like, run. So
we see that a lot, Chris. We do see that too many times.
I know those people exist, but
Yeah.
You know, you have to have humility. If you work in the field of oncology and you are not humbled by how difficult it is, you are incapable of being humble, which is a huge problem, I think. So yeah, I think that's a great piece of advice that I would recommend. You know, that could almost be like a core tenet of One Cancer Place. Like this is something you should never miss out on. If you go see your doctor and they don't want to work with experts in the field, you probably need a different doctor. Like
I agree. I agree. And I hope that is the singular part, the core message to the new patients that we start developing and talking to and gaining trust with is exactly that. And I would like to be able to provide a way, a mechanism by which they can have that second opinion very easily.
Yeah.
Right then, I believe that starts the wheels turning on education and trust.
Yeah.
And that's really important as we work with patients at such a tender moment.
Yeah.
Oh my God, this is such a time for patients that.
So if One Cancer Place became a like a first stop for people newly diagnosed with cancer, right? Like it wasn't about your specific tumor and all these decisions you have to make, but just more about some central guiding. Here's some things you need to know before you get started, right? Boy, I think that would be on the top of my list of something to start with. Just like Julie just said, like there are experts out there who are more than willing to help you, and all you need is a doctor who will engage with them, right? I think those are really great starting points. I have to go to a meeting.
But thank you so much, Chris, for doing this with us. I hope you'll do it again.
Yeah, absolutely. Thanks for having me.
I mean, it was fabulous hour and a half with both you and Dr. Flaherty, and I'm sure everybody joins me in saying thank you, thank you, thank you.
Thank you.
Thanks for having me. I feel like I learned something every one of these conversations, so thanks.
Let's talk soon.
All right, bye.
Thanks.
Thank you.
You guys want to stay on a little bit? I think we can for a minute. Danielle Monterey, did you enjoy?
I enjoyed it.
That was very high level stuff, high level cancer science, and a lot of words thrown around, a lot of phrases thrown around that we don't hear that often, but we get more and more used to hearing the more we're in the conversation. And I'm shocked at my understanding these days of some of this, and it's really only based on experience and having been around the conversation. So don't be daunted. It is like they do speak another language, and I'm always totally shocked when I realize that I'm understanding what they're talking about. So don't be daunted by all of this, and keep on the learning path, because I tell you, it's really, really worth it. And we really, really can help other patients, and they really, really want us to. So Ellie, I'm so glad to see you.
Yeah, I'm glad I was able to make it.
Yeah.
That was wonderful.
Wasn't it wonderful? Now here's what we're going to do.
We're
going to edit this, and we're going to put it in the learning library or learning lobby library on the website. And this is going to be the first of the content for that library. So people will be able to go back to it. And I'll bet you all of you are now going to go back and watch that interview with Peter Attia that Dr. Flaherty did.
I watched it once, but I need to watch it again.
I have listened to it 4 times. And on the 4th one, I finally heard one of the most important new learnings for me that has all the application to what we're doing here. And that is that 90% of the scientific drug development going on right now is under wraps with little small biotechs that have one molecule they're researching. They haven't even gotten to phase 1 trial yet, so they don't know if it works. But think of this, 90% we don't know about. Now, Dr. Flaherty's business, his molecule that the company called Loxo, he sold that to Lilly. And but it started with one I don't know the name. I think the name of the drug you probably know, Julie, Aricipta or something. I don't know. And I don't know that I think it treats BRef and MEK. I think that's his specialty thing. But that was and that point that he made in that interview that I listened to for 4 times, I finally got on the 4th time, was that 90%, there's 90% of drugs being, that we don't know about. 90% of 100% is still unknown. And if that isn't hopeful about what we do, I don't know what could be. I had no idea. I thought maybe 10%, maybe 12% were in development. I had no idea because you don't hear this conversation. So when I heard that and got it, I went, holy moly, that's really something that he's talking about. And I've got a story that illustrates this that I want to share with you. And that is a story of a patient, a woman who was in the clinic professionally, and she was diagnosed with cancer. I think it was breast. I don't remember which cancer. Anyway, her husband was also in the clinic, both professionals. She's diagnosed, and they decide to do everything that they know against finding a cure for her. And what that journey did for them was they found something, I don't know, something to follow, some new drug to follow in Chicago. So they went up to Chicago. Then they went to Fred Hutch in Seattle for another one. Then they went down to San Diego, UCSD, and met with that doc down there, Dr. Razzouk, who's really front and center with a lot of the new thinking and drug development and treatment. And somehow they found out about a little, by this time they knew the tumor at her tumor at the molecular level. And they realized that they had either an ERK or a MEK pathway problem. Whatever that means, there was a problem. And there was a little company in Kansas City that was developing a molecule to address that little problem, MEK or ERK, whatever it was. So they went to Kansas City and they got that little drug under compassionate care, and she's now NED. And she started out as stage 4, whatever her cancer was. Now, if that isn't going to the ends of the earth to find your molecule, I don't know what is. But I told Dr. Flaherty that story, and I said, this is the sort of story that we need to know about in One Cancer Place, and we need to talk about in order to encourage education and connection and talking and finding out everything you can and encouraging hope and never giving up, basically. And so, I mean, I tell you that story because it just gives me so much hope. And then hearing that there are so many companies out there in this same situation as that little company in Kansas City, Kansas. I think they're in Kansas and not Missouri. Anyway, that's my story. And I hope you guys find it helpful. I certainly do. Mary, what do you think?
And I'm quite amazed at what I don't know. I mean, I knew I didn't know.
Well, that's our story when we go in. We don't know what we don't know.
Exactly.
That's what makes it so unbelievably difficult for, to reach. It makes it really, really hard to help a new patient. It really does. And, we got a huge challenge in front of us. But I heard second opinion and decision support as big chunks that I think we could go back and talk to Dr. Flaherty and Dr. Heery about and get a lot of support for, obviously. So we're in a good spot. I'm really glad that you guys came today.